So once the tetrad forms and crossing over occurs between homologous chromosomes, the next thing that's going to happen is that the homologous chromosomes as pairs move to the middle of the cell. So they're going to move to the middle of the cell as a pair and line up like this. Now, this is important. How they align along the metaphase plate is different each time. Now, in humans, here I only have 3 pairs, but imagine in humans, there would be 23 pairs of chromosomes lining up down the middle of the cell. And there's so many different possibilities of how this could happen, but that's in a later slide. So here you have the homologous pairs line up along the middle of the cell, and then they're going to get separated. Now, I don't have it shown here, but spindle fibers are pulling apart the homologous pairs. And then you have cytokinesis. So at this point, this was the first part of meiosis, and we called this well, that's not writing. Hold on. The first part of meiosis, and we called this M1, or the end of M1. So everything we've been seeing so far is M1. Okay. And then we have cytokinesis. Now, at this point, I want you to think for a minute. Are these cells haploid or are they diploid? Right? Do they have one set of chromosomes or do they have 2 sets of chromosomes? Right. They're haploid. They have only one set of chromosomes. Remember when we started, we said 2n equals 6 and n equals 3? Well, there's only 3 chromosomes in each of these cells. So at the end of meiosis I, the first round of division, we technically have haploid cells. So how come we can't stop meiosis here, right? Like how come we can't just say, cool, haploid cells, we're done? Because at this point, we still have duplicated chromosomes. Our chromosomes are still in that like that X shape. There's still sister chromatids attached by a centromere. So here, how come meiosis isn't finished here, especially if the cells are already haploid? That's your box 4. So now our next step would be to separate these sister chromatids so we can have cells that have n equals 3. So now the next part, this is now technically M2. So we're going to divide our nucleus again. So here the chromosomes move to the center of the cells. And just like we saw in mitosis, they're also going to get separated. The sister chromatids are going to get separated to opposite ends of the cell. And then we have cytokinesis. Now I'm just going to make that a little bit easier to see. Now when we look at these cells, we can see that none of them are identical. At the end of meiosis, we have 4 haploid cells that are all genetically different. And one of the main reasons they're genetically different is that crossing over. Because crossing over occurs, we've now switched pieces of our DNA and we have no identical gametes. They're all different. They're all unique. And now this is only with 3 chromosomes. Imagine if we had 23 in each of these, all the different combinations that would happen after each round of meiosis. Now, if we're forming sperm, they'll actually go through processing where they're going to add tails to the end of the sperm. So here would be gametes forming sperm. Now, if these were females, what would actually happen is you're going to end up with only 1 actual egg and then 3 polar bodies. So when we compare and contrast meiosis in males and females, you still go through 2 rounds of meiosis, meiosis I and meiosis II. You can see meiosis I is going to be separating homologous chromosomes, and in meiosis II, we separate sister chromatids. Now at the end though, or you can kind of see here in this first round of division in forming eggs, that the cytoplasm is not evenly divided. One cell kind of hogs the whole cytoplasm. So as it divides again, you can see this one is little, and then as these divide, one of them is going to take the majority of the cytoplasm. These 3 polar bodies are all genetically different. They have the results of crossing over in them. However, they're going to disintegrate and be broken down inside the female, so she ends up with just 1 egg. So at the end of meiosis, a female gets 1 egg and a male gets 4 sperm. And this is why we generally have 1 offspring. Now, if you're having twins, that would be meiosis happening twice, and you get 2 separate eggs and 2 separate sperm to fertilize. Or it could be that you had 1 egg, 1 sperm, and then that divided, and now you have identical twins. But we can talk about that more in class. All right, so here's just another picture kind of showing how you start out with diploid cells, 2N, and throughout meiosis, meiosis I. These cells right here have the sister chromatids still, so we would need to separate again in meiosis II. And you can see how the unequal division of the cytoplasm happens in the females to result in 1 large egg, which is the largest cell in the human body. It's the only cell we can see without a microscope, like with just the naked eye. Okay. All right, so I want to point out another source of variation and something that's kind of cool during meiosis. So here we're back in interphase. We have our 6 chromosomes. And remember when they duplicated and then when we entered into M phase, how they like pair up and find the homologous pairs? Now this process here is random, like how they end up pairing up. Like when I say random, so here is a little cleaner view. What I mean is, pay attention to these chromosomes right here. Couldn't it have very likely ended up like this? Absolutely. Now, how the chromosomes end up when they pair, oops, wrong way, when they pair is random every time. Which side the chromosome coming from the mom and which side the chromosome coming from the dad comes from. So when they move to that middle of the cell, you could have this combination or this could have happened. This was in my original slides of meiosis, but if I had very easily, I could have put the orange one on the left. Then I'd have different, when these go to divide, when these separate, into independent daughter cells. So like here, when cytokinesis occurs after meiosis I, and these become into a cell and these become into a cell, I'm going to get different gametes than I did the first time around. So independent assortment is another way that we can get variation in the offspring. When you look here, so if we have these 4 chromosomes, 2n equals 4, you can see after meiosis I, then here I have meiosis II, there's different possibilities of how the chromosomes could end up together. If you look at this stage here, there's different combinations. And this is only with 2n equals 4. Imagine 2n equals 46, all the different possibilities of them lining up along the middle of the cell. So independent assortment is one aspect of sexual reproduction that generates genetic variation. And it's the random orientation of pairs of homologous chromosomes at metaphase of meiosis I. So this part here, it's random. Like you can see the difference between these 2 cells, how on this side you have the purplish pink one and on this side you have the blue one. That totally changes what ends up in the gametes. So that's independent assortment, how they assort along the middle of the cell. Each chromosome pair is independent of the one below it. They don't influence each other whatsoever when moving towards the middle of the cell. Okay. So how does independent assortment contribute to variation in offspring is your box number 5. And then there's something called random fertilization. So with random fertilization, I believe in males, they make about 100 million sperm per day. And so during sexual reproduction, which of those sperm actually fertilizes the eggs is totally random, right? So you're going to have so many different combinations of genes end up in those 100 million sperm. Which ones actually fertilize the egg is a totally random process and could have any combination of traits in them. So when we talk about random fertilization, that's another way in sexual reproducing populations that you get variation, because every time fertilization happens, it's going to be a different combination of genes because of crossing over, because of independent assortment. You're going to have different combinations of genes get fertilized or form a zygote every single time fertilization occurs. If you get rid of crossing over, you don't even talk about crossing over and you only talk about independent assortment, there are 8 million different ways, different combinations we could put those 23 pairs along the metaphase plate. So right there's 8 million different combinations possible every time meiosis happens, and you add in crossing over. There's so much variation in each sperm. It's all unique, and it's random which one fertilizes that egg. So if I were to have 10 kids, all of my 10 kids would be unique, unless I had identical twins. Yeah, they may look similar because they're coming from the same parents, but they would each have different combinations of genes. So when we look at the big picture here in meiosis, or one of the big pictures, besides making haploid cells that are all different, are the 3 main sources of genetic variation. Now this is different than mutations. This is just talking about sources of variation in meiosis. Now one of them, our first one, was crossing over. Crossing over literally breaks off chromosomes and shuffles them and moves them around. And we have independent assortment as another source of variation. How those homologous pairs line up at the metaphase plate can be separate every time, or separate, can be different each time, and they don't influence the homologous pair below them. And then we have random fertilization. It's random which sperm fertilizes the egg and which combinations of genes are going to be forming that new individual. All right, and that's your box 6, and we're all done.